must say I was particularly impressed by the list of the previous recipients of the award. Some of them I know them quite well, and they have been role models for me. Some of them, as I was, a young developmental neurobiologist, so to speak, they were already quite established. So to me, I feel very honored, obviously, to join this, prestigious list of developmental neurobiologists, and I'm very honored by that. I also feel very grateful, and I think a lot of my lab members, whether the members in my lab currently working in the lab or who used to work in the lab, because obviously without them, none of the research that I've been talking about and for which I'm lucky enough to get that prize would have been possible. There is a Nobel Prize winner of my country, Belgium, Christian de Duve, who discovered lysosome almost half a century ago, and he was once interviewed and he was asked, what is it that made a difference in your career? And the journalist, I think, was asking, do you think it was because you had great mentors? And he replied, yes, indeed, I was very lucky to work with very good mentors, but most importantly, I was lucky to work with extremely good students. And even though I really don't want to compare myself to Christian de Duve, I do completely sympathize with this notion that on the one hand, great mentors are a great way to start doing science, but in the end, the people who work with you, that you train and who actually make the discoveries in your lab, that's the most important people in one scientist's career. And so these are the ones to who I say thank you, and with who I want to share this prize as much as I can. So in my lab, we are studying the mechanisms of development of the brain, and in particular of the human brain, and of a structure in the human brain that is arguably the most complex structure among all living organisms, and that is the cerebral cortex. And we try to understand at the molecular and cellular levels how the complexity of the cerebral cortex arises during development, also in link with the evolution of the human brain, because the cerebral cortex is one of the most, as we call it in evolutionary biology, divergent structure, meaning the most special structure, if you will, in our brain. And we try to understand which mechanism development has changed during the evolution of great apes and in the human lineage, and that help explain the specificity of the human brain, such as the increased size of the human brain, and most importantly, the increased complexity of the human brain that makes its function so characteristic. Mm-hmm. Yes, so that's a tough question. That's really the question we try to understand. So, in short, the roots of the differences are in differences in the development. So that means that the way our brain develops displays a number of differences that explain how our brain then, in the end, becomes different. One very striking thing is that our brain is larger than the brain of all other great apes. And it's larger mostly because it has many more cells, in particular many more nerve cells, which are basically the building structures of our brain and of its function. And one of the reasons why we have more nerve cells is because the stem cells of the brain, the neural stem cells of the brain that are active in development, display particular properties that enable them to amplify more and to generate more nerve cells. And one of the things we do in the lab is to understand at the molecular level what's different in our genome that makes the stem cells of the human brain able to generate more nerve cells so that our brain becomes bigger. But the bigger difference, in fact, is beyond brain size, is in fact the way nerve cells are connected with one another, because the way nerve cells are connected to one another is really going to define how the neural circuit, I would say the neural circuits of the brain actually work leading to specific types of perception, thoughts, and behavior. So this is really the neural circuits that make us what we are as individuals and as a species. And so this is really one of the mysteries that we and others, I think, are very eager to try to solve in the future, is to, first of all, understand what are the mechanisms that control the wiring of the brain, so how nerve cells get connected with one another, and then also how there are differences in the wiring between different species that help explain how is it that we have properties such as abstract thought, long-term planning, or language use, for instance, that are so uniquely developed in our species compared with other species. The one thing, though, that I think is worth mentioning is that it would be unique about the human brain. Evolution is a continuous process, and so there are actually more and more properties that scientists and people in general thought were unique to the human brain and that are in fact shared with other species, whether great apes or other species. So it's becoming actually more and more difficult to define exactly what makes the human brain unique, but let's say it displays a number of qualitative and quantitative differences that, for instance, enable me, to have this chat with you today, and for you and the listeners to understand, hopefully, what I'm talking about, and that I think we do to a degree that no other species is able to do, because the nerve cells in our brain are wired in a unique fashion, and seeking to understand how this wiring takes place is really, I think, a key question for the future for our lab, and I think for many other labs interested in brain development. So mitochondria are small organelles, small structures in the cell, in every cell of the body. And they are thought to mainly serve a very important function, which is to provide fuel to the cell, energy. Now, what we found is that during neurogenesis, they do much more than that. They actually send signals to the cell to basically change the fate of the cell. Indeed, what's important to know is that neural stem cells, in the process of neurogenesis, each time they divide, they have to decide, basically, to speak in an anthropomorphic way, they have to decide whether they remain a stem cell, so they self-renew, or they become a nerve cell and they differentiate. This is a very important decision. And this decision is helped by the influence, is influenced basically by mitochondria that provide signals, and we don't know exactly yet what is the molecular nature of these signals, but we know these signals exist. They come from the mitochondria, and within the cell, they tell the cell, you will self-renew or you will differentiate. So this is very surprising, because usually it's a completely different class of processes that control neurogenesis and that control development in general. And so now our findings are basically part of more and more findings made by a number of other colleagues, other labs in other fields, that mitochondria and more generally metabolism of the cell is actually not just, let's say, a permissive process just to keep the cell alive, but also an instructive process that can influence the cell and direct it to become another cell or to change completely of state. So that is, I guess, the new thing about our findings of the influence of mitochondria on neurogenesis. There's a second observation that we made that is very intriguing, even though we don't know yet, let's say, all the answers. And that is that the mitochondria instruct the fate of the neural stem cells at a very defined time period, a period that we call a critical period, a period during which the cells may decide to self-renew or to differentiate. And this period has a different length if we compare mouse neural stem cells and human neural stem cells. And to put it in a very simplified way, I would say what we observe is that human neural stem cells take much more time to decide whether they will become a nerve cell or self-renew as a neural stem cell. And this timing, we think, may be actually crucial and provide more potential of amplification for the human neural stem cells, which is indeed a property of the human neural stem cells, is that they can amplify more. And this is one of the reasons why we think that the human brain is larger than, for instance, the brain of the mouse, is because the neural stem cells can expand more. So it could be that there are special properties in the, let's say, the dialogue between the communication between the mitochondria and the cell that is different among different species, so that this timing is different, so that in the end we get a different neurogenesis and perhaps a different brain size. But of course, this latter part that I said is still speculative, and this is really one of the things that we want very much to study in the future. But of course, if it is true, it would be quite remarkable, because mitochondria are very old structures. They are present in all cells of living organisms, animal or vegetal, and yet it could be that they may have played a little role in the evolution of the human brain. So that would be, of course, quite remarkable. But again, more work will be needed to test that. Yeah, so this is a very interesting question. So we have started to do transplantation of nerve cells that we can generate from stem cells. So we've started these transplantations basically as a tool to study brain development, to study the development of nerve cells. And that was almost 15 years ago. And if you had asked me at the time, will this ever lead to, interesting prospects for brain repair, I would have been very cautious, and I still am, but I would also have been quite skeptical of, let's say, the prospect of neural repair with stem cell therapy. But I must say that the observations that we made, and that actually many other colleagues made, that when you transplant a young nerve cell into an adult brain, these cells seem to be capable to integrate in a very fruitful way, very physiological way, very functional way in the adult brain, is very surprising, and it opens very interesting possibilities for brain repair. Now, whether this will be used to treat patients in the future, that remains the big open question. And so I think one of the important thing to say is that it depends on the disease. So for instance, for Parkinson's disease, that is thought to be caused by the loss of specific types of neurons, relatively few of them. There are actually clinical trials ongoing using stem cell derived nerve cells and their transplantation in the hope of treating these patients that suffer of Parkinson's disease. And those clinical trials that are, as I said, that are just starting, will be really crucial to validate in a clinical setting this concept that you can repair lost cells with new cells in the brain. But I think the experimental work that has been done so far is promising enough that we can hope that perhaps it could work. Again, the clinical trial will tell us. As for the cerebral cortex, so the structure that we are mainly interested in and that we study, I think it's a different story, because the cortex is, as I told you, the most complex structure in the brain. It contains billions of neurons connected with trillions of connections with one another, and they have to be connected in a highly specific way. So whether we can reconstruct, let's say, part of the motor cortex after a stroke will require, something much more complex than what we can currently do. And so the jury is really still out whether this will actually happen. I think in any case, this is going to take, I would say, decades of research before we can actually do this in a fruitful way. But this is not a reason why we should not do the research. First of all, because even decades of research could be, of course, very useful even though it's not for us, but maybe for our grandchildren or something like that. And on the other hand, this research is also important because it reveals very interesting insights about how the brain, the adult brain, and the neural circuits in that brain can react to new environment. For instance, the appearance of new neurons in that circuit. That's very related to what people usually refer to as neuroplasticity, which is basically the ability of the adult nerve cells or brain cells to react to the environment, to react to changes. Sometimes in a way that is actually making the whole circuit work better, so to speak, in a more adapted way to the environment. And so how transplantation of new cells, disease of old cells, and plasticity are linked, I think could be a very interesting target to study to improve neurological conditions. What I mean by that is that perhaps what we study now on the interaction between young cells that we transplant and the old brain could be also important and could provide new insights to treat neurological conditions even without transplantation, by understanding, in particular at the molecular level, what makes a diseased brain, what makes it able to adapt to that disease and work in a way that is functional again. And that is, I think, a very interesting and very open question for which developmental neuroscience can bring a lot of interesting knowledge in the years to come. Yes, so I would almost say for personal reasons, meaning that my father was a scientist. He was actually a neuroscientist and a neuropathologist. And so I was lucky actually that thanks to him, I could do a lab internship at the National Institute of Mental Health in Bethesda, next to Washington, for a whole summer. And this was really a revelation for me. I was a young medical student. I liked basic science, but I didn't really know what to do. And to spend a few months performing experiments by myself, trying to get data. I didn't get that many data. Most of my experiments miserably failed. But I really got caught by this. And I really realized how science can become a passion. And that's basically what happened soon afterwards, as I started to work in a lab, first as an undergrad student and then a PhD student. So it's this summer break internship that really was a turn point for me. And I often say actually to students who start in the lab that indeed the only way to know what research is really all about is to be in the lab and perform experiments. You can read books about science as much as you want, but the real thing is the practice of asking a question, trying to perform an experiment, and then try to understand in all the data that you gather if there's any sense. This is really, I think, what science is about, and the only way to find out whether you like it or not is actually to do it.